Okay, thank you for coming, everyone. Today, I'm excited to introduce our speaker, Sam Morabito from the swROPE Lab. I've been watching him work on this software that he's been developing for the last few months here, and I'm very excited to finally have him share his story and these really neat tools with all of you. Yeah, I guess take it away, Sam.
Okay, thanks, Fairly. So I'm going to talk about single cell WGCNA. First of all, what the heck is, I hope you all know what single cell is, but what the heck is WGCNA? So it's actually like this, at this point, like ancient way of analyzing gene expression data. And specifically, it's looking at co-expression networks. So these are kind of like some of the first publications about WGCNA. And if you look at the dates, it's like 2008. Some of them are even like 2006 and 2007. And it seems like the authors of this method, pretty much like every single piece of it was its own paper, which is crazy. But it's actually like, even though it's old, it is still a good method. It's actually been cited over 10,000 times. So people, and it's still like getting lots of monthly downloads. So people are still using it all the time. But there's actually like, so it was designed when we were doing bulk gene expression. So taking gene expression measurements in an entire sample and not at the single cell level. And there's a lot of considerations that you actually need to take before trying to do this kind of co-expression network analysis in single cells. So this is a figure from the WGCNA paper that kind of goes through the like overview rationale of it. The first step is essentially to take a gene expression data set and construct a gene co-expression network. So it's kind of considering, it's basically a network between different genes as the nodes, and the edges in the network are some relationship between those genes. And yeah, WGCNA is using correlation as a measure of co-expression. So the edges between genes are their correlation in expression. So genes with high edge weights are basically genes that are expressed in the same samples. And then there's also can be genes that are like anti-correlated. So using this kind of adjacency matrix that we see here, you can do a clustering algorithm. So they have something which is its own paper called dynamic tree cutting algorithm that does a hierarchical clustering, like you see this dendrogram, and then it determines what they call like gene modules, which are modules just essentially another word for cluster. So it takes this hierarchical clustering and then it does the dynamic tree cutting algorithm to give us these distinct gene co-expression clusters or modules, which I'll be referring to them as. The next thing in the WGCNA workflow is essentially to figure out what are these modules. Because this is an unbiased approach, we have to kind of determine what the functional role of each module is. So this is done using like enrichment analysis and correlation with different clinical data or other types of omics. And you want to basically find what are the biologically interesting modules. So for example, like if you have, if you're thinking about like control versus disease, you might want to figure out which modules are highly expressed in disease versus control. The other thing you kind of want to do is study the relationship between each module. So like which ones are correlated with each other, which ones are anti-correlated. And the last thing that the original WGCNA does is try to find the key drivers in these modules. So it's like looking at the network structure to figure out which genes are the most highly connected in the network. And then kind of thinking about those genes as like potential biomarkers for like identifying a disease or like using that, trying to modulate that particular gene as a treatment of the disease. Anyways, so we took, when we started to generate our own single cell data sets all the way back in 2019, we wanted to figure out a way to use WGCNA on our own data. The one thing that we noticed at first was that the sparsity of single cell data is an issue when trying to construct a co-expression network. So we've looked at the literature and saw that others were taking this like Metacell aggregation approach to do network analysis in ATAC-seq data. So we kind of took a similar rationale to aggregate groups of very similar cells into metacells before feeding it into WGCNA. And in our original approach, we also included bulk RNA-seq data in like as well as the metacell single cell. So we have these two different cohorts of bulk RNA-seq, and we have two different cohorts of single cell RNA-seq. And then we performed WGCNA on that. And we did it iteratively for each cell type, because each cell type is like in single cell, we know that like a lot of the variation is driven by the cell type identity. So it'd be harder to tease out the differences between control and disease in the network if the correlations are if you perform the correlation network on all the cell types put together, basically. So for that reason, we kind of take this iterative approach and do it separately for each cell type. So this is just a figure from the paper that is already published. So, and I know this is a lot to take in, but basically each of these these like circles with like all these letters and like lines everywhere, each of these dots is going to be a gene, and they're labeled with their gene symbol. And then these are actually the hub genes, so the most connected genes for 4 of the WGCNA modules in the oligodendrocyte cells. And if you're familiar with oligodendrocytes, some of these are basically myelination marker genes. So like myelination is the main thing that oligodendrocytes are known for. So these CNP and PLP1, MAG as the marker or as the hub genes are not really surprising. Tells us kind of like, oh, this is a myelination gene module. And one thing that we did with this was correlate the module eigengene with AD diagnosis. And we found, okay, this green module 4 is actually correlated with Alzheimer's disease. And the other ones are correlated, but they're going like in the opposite direction. The other thing we did is in this paper, we kind of, using our different analysis, we had focused in on a particular transcription factor we were interested in. So we wanted to see, is there an overrepresentation of the target genes of this transcription factor in any of these modules? And in 3 of them, yes, there are target genes. So potentially this SREBF1 is a regulator of some of these modules. So you can already see that like WGCNA is telling us kind of these things that we can't really get from other analysis in single cell, such as like differential expression doesn't tell you anything about the relationships between each gene. It just tells you about that gene on its own, right? So anyways, there's a few shortcomings of this original approach. Yeah, what's up?
I have a question on the previous slide.
Okay.
So for SREBF1, is that just a transcription factor that you looked for because of prior knowledge that you had about it? Or did you find that it had some interesting, like, was it in one of those modules and that's why you looked at it? Like, is there, I just want to know.
I think I said it, but we had like, so this paper was like, the scWGCNA was like a tiny part of it, basically. It was mostly an ATAC-seq paper. And we had identified SREBF1 as interesting based on our ATAC-seq data analysis.
Okay, so you found like differential peak accessibility or footprint accessibility.
Yeah, and like, yeah, exactly. So
okay,
it was like kind of like lucky that we identified that, because it's been since implicated in other like totally independent studies of Alzheimer's. So that was cool. But anyways,
cool. Sorry, I missed that.
For sure. There's some shortcomings of the original approach. Basically, like I did this really early on in grad school, and like it was really like hacked together essentially. It only contained the most essential functions, and everything else was just like code floating around, like various scripts that I had. So as it turns out, I had to like, this wasn't the only single cell RNA-seq data set I analyzed to date. I like with every single data set I got or have gotten in grad school so far, I've had to, eventually someone asked me to run scWGCNA on it. So, and every time I did it, it was kind of painful. So at some point I was like, oh my gosh, I have to make this better. So very recently, like in the past couple months, I've made like basically major updates to scWGCNA to maybe make it usable. I don't think anyone, this is like very new, so I don't think anyone's actually tried it. But yeah, essentially, like now I would describe this, like I used to describe it as an add-on to WGCNA, but now I would say it's like its own package, and it's an extension of Seurat and WGCNA. So it works entirely on Seurat objects, and I'm trying to take a similar design philosophy to Seurat, because I would say Seurat is like pretty easy to use even if you don't have coding experience. So I kind of wanted to make scWGCNA like that, so it's pretty accessible to anyone who needs to do this analysis, not just like, and I think like previously with WGCNA, it's kind of like it's much harder to figure out. A lot of the plotting and stuff that you see with WGCNA are not, they're like, it's all custom code. It's not functions that they give you. There's just like tons and tons of settings for WGCNA. And I tried to like pare down the amount of things you can change here. So I think I'll go through like how, basically the next part of this talk is going to be like going through how you would analyze a single cell data set with the new scWGCNA. So there is this full package down documentation site that I'm working on. It's not done yet, but it's I think that the key features are there. So it'll tell you how to install it, and it'll tell you how to run it, basically, if you look at the vignettes section. So yeah, now I'm going to go into like how do you construct a co-expression network starting from a straw object. So yeah, like I said, scWGCNA kind of starts where Seurat ends. So this is on the right. I have just a list of the Seurat functions that kind of take you from start to end. So for, I mean, before you even normalize, you would do your quality control, and then you do normalization, feature selection, scale the data, batch correct if needed, run PCA, run UMAP, and then perform clustering. And then optionally, you can annotate your clusters as cell types of interest or whatever. So this is the single cell RNA-seq data from my published paper. But we're actually going to use, I don't know why I always like this data set for testing things on. So it's this other human Alzheimer's data set. But just to kind of make things simple for testing scWGCNA, I only am including control brains here. And also to kind of prove that it doesn't just work with Seurat, I actually analyzed this whole data set with ScanPy, and then I converted it to a Seurat object. So yeah, I wrote down a little bit about how I process this data with Callisto, and then Cellbender, ScanPy, Harmony. Okay, so I'm going to show these like little blocks of code just to see like, just kind of show like, how would you do these things with WGCNA? So the first thing you have to do, like I said, is to compute the meta cells from the Seurat object. So I guess before even doing the first thing, you have to run this setup function, which is just making a spot in the Seurat object to store everything that we're going to do with WGCNA. So it's just this one simple function called setup for WGCNA. It actually also will pick which genes you use for WGCNA. You typically don't want to use all of the genes in your Seurat object. You just want to use genes that are going to be expressed. So the default behavior is basically it will take genes that are expressed in 5%, at least 5% of cells. You could also give it just the variable genes. You could just give it marker genes, like whatever you want. We're also just giving it this name for the WGCNA experiment. We give it this name essentially because you can, so like I said, you can do scWGCNA iteratively for each cell type. So you might actually end up with one Seurat object that has like 5 or 6 different WGCNA experiments in them. Okay, so after we set up the Seurat object for WGCNA, we run the metacells by groups function. So this is actually constructing metacells separately for each, any like groups that you give it basically. Typically, we want to do this split by biological sample, and we want to do it split by cell type or cluster. And then we also give it the k nearest neighbors parameter. So here is just k equals 25. So that's basically for, that's like how many cells are going to be aggregated together to form one metacell. And then this function, it's, I don't know, it's not like the fastest function in the world, but it'll probably give you results in like 20 to 30 minutes, and then you can kind of be on your way. And this is kind of like an optional thing, but if you want to see like what does my Metacell data set actually look like, you can kind of analyze it. We actually store it as its own Seurat object, and then you can run the regular Seurat pipeline on the Metacell data and then visualize it with UMAP. So that's what you see down at the bottom. And you can see that we are retaining the information about which like biological sample on the right, and then which cell type it came from. And it doesn't look it looks quite similar to a single cell data set. Like if I just told you this was a single cell data set, you'd probably believe me, but it's actually the aggregated metacells. Okay, so I'm just showing this plot because of I said that we do this metacell thing to alleviate the problem of sparsity, and just so you believe me, I'm plotting the density of each expression matrix split by cell type for different levels of for different numbers of K. So this is just like the K is how many cells you're putting together, and then all the way on the left side is just the raw counts matrix, and then all the way on the right side is the full pseudo bulk matrix. So that's putting like all the cells from the same cell type together. And this is actually plotting density, but and density is just 1 minus sparsity, where sparsity is defined as the number of matrix elements that are 0 divided by total. So you can see here the actual counts matrix for like the inhibitory neurons. This is basically telling us that like only 10% of the elements in the matrix roughly are non-zero. And then when we start using metacells, like if we use K equals 25, which is what we actually used for the analysis, it's like the density increases like 4 or 5-fold. So that's going to end up helping us downstream in the actual co-expression inference. Okay. So
Sam, you had a question in the chat.
Oh, I'm sorry.
That I also had too, which is what's going on with the missing OPC bar?
Yeah, that's actually like I put that there on purpose because this is what happens when you run, like you can give like any number to K, but what if like K is larger than the number of cells that there are? Then it just doesn't work. Does that make sense?
So there were less than 100 for OPC.
Yeah, so basically like, what is it? Since I'm splitting it by each sample, that means like, since OPC was one of the smaller clusters anyways, there's probably fewer than 100 for like one of the samples, or maybe even all of them. So this is kind of to help you, like, I don't know, figure out which number of K you can use. I typically always use 25 or 50. I think on the website I recommended to keep it between 25 and 75.
I have a question.
I go for it.
If do you set a parameter for the number, for like the percent overlap between the metacells?
Do I? I don't think I do, but that is something that could be done. But currently, no. We do like I think like we plot, or not plot. We like report as outputting like the median number of cells that overlap, and it's pretty much always 0.
So the median number is almost always 0, meaning that there's no overlap between the two metacells.
There's gonna be some overlap, but it's like pretty minimal.
I see. Because I don't know if you do want overlap, or because if you don't have overlap, then you might have cells that aren't captured in any metacells, right?
I haven't looked at if that actually happens, but I think that almost all the cells are getting captured.
I see.
I maybe want to look into that.
And if
That's a good point.
if K is too low, then no, then that's not okay. Thank you.
Yeah, so I guess there's a couple of problems, like well, if you want your downstream steps to run quickly, you'd want like to have a smaller data matrix. So if you use a really small number for K, you're going to end up with more meta cells.
Mhm, mhm.
So I don't know. Just like, just by using this a bunch of times, I've pretty much like only used 25 or 50.
What if the data set is much larger? Would you increase K or should you just stay at 25 to 50?
So it actually depends more on like, if you do it this way, like I did, where you split it by cell type and by sample,
Mhm.
even if your data set is really large, each sample will probably still have like a similar number of cells. So I guess, I mean, you could just not split it by sample. Like, let's say you just split it by cell type and then like control versus disease condition. In that case, you could increase the number of meta cells for sure.
Okay. Okay, thank you.
Yeah, for sure.
And then there's one more
Um,
question in the chat. Sorry,
yeah.
Andrew's asking if this plot is like easy to make as part of scWGCNA, or if you'd have to write custom code for it.
Okay, I have the code. So I was planning to include this plot in the like scWGCNA paper, but it's actually not currently on the website. But I can definitely add it if this looks interesting.
I think it would be useful.
Yeah, it takes a while to run though, because you have to like compute the meta cells like over and over again. But I mean, yeah, I don't know. It's
It would
definitely
have some
interesting.
utility. I mean, perimeter
Yeah.
optimization is like a huge problem in single cell. So it'd be nice to have a way to
Right.
like confidently pick your K. And then also I'm going to ask you a question, Sam, that I asked you in 2019 when you first were talking about this, which is, would you ever want to scale K based on the number of cells that are in your cluster? Or in your like, you know, the number of cells that are in your like sample cluster
Yeah.
combination?
I see what you're saying, but I'm like not sure if like, okay, like technically you totally can do that. And I guess like, I don't want to put the cart before the horse, but like, you're gonna like be doing WGCNA like separately for each cell type. So let's say like in this data set, we want to do it for all 6 cell types. You can do like, you can just use this, use the same meta cells, but if you actually want to use a different number for each cell type, you totally can.
Okay, cool.
Yeah, I just haven't done it that way, but like the software does allow you to do that for sure.
Cool, thank you.
Yeah, okay. Anyways, there's like one more parameter that is like really important for WGCNA, which is called the soft power threshold, or beta. So I didn't want to talk too much about math, but like this equation is actually the formulation for the co-expression network. So A is the co-expression matrix, and then I and J is like the pair of genes I and J, right? So the value of the matrix A for pair of genes I and J is this absolute value 1 plus the correlation of the expression between I and J divided by 2 and then scaled to a power beta. So we've scaled it to the power beta because WGCNA tries to make a network that has a scale-free topology. And a scale-free network is basically a network whose degree distribution, degree meaning the number of edges coming out of a certain node, so the number of genes that each other gene is connected to, the degree distribution should follow a power law. And there is a very, very long Wikipedia article about the implications of a scale-free network, so I don't really want to go that much into it. I just want you to know, okay, this is an important parameter to tune. So there's this whole step where we sweep for the beta value, and then we actually fit how well does our network follow this scale-free topology. And then just the rule of thumb that the authors of WGCNA give us is, okay, we want to pick the beta value where the scale-free topology fit is 0.8. So if you look at this curve, we follow it, okay, where's 0.8? Okay, the first value is 9. That beats 0.8. So then our value we pick is 9. And that's what it is. So this test soft powers function will give you, it'll sweep the parameters and it'll give you this plot as an output. And then we're finally ready to construct the
Okay,
co-expression
hang
network.
on.
So
No, we're not, because we have another question. Axel is asking, why should
Honestly,
WGCNA, oh, sorry.
I don't even want to get into this question because this can take the entire time.
Okay, so perhaps we need another separate GenPALS on just WGCNA.
I mean, we could have a journal club for WGCNA that lasts the entire quarter because there's so many different papers on it. So I was just like, I mean, at least for the purpose of this talk, let's just assume that we trust them, that it needs to follow the scale-free topology. Anyways, so there's this construct network function, and that's all you really have to do to construct the network. There are a bunch of different parameters that you can give to it, but the only one that you really need to tune is the soft power beta threshold. So here I'm just saying 9, and then it does its thing for a while, and then you can kind of make one of these, like I was showing earlier. The WGCNA dendrogram is kind of one of the main plots we look at. So each each like leaf in this tree is a gene, and then you can see the module assignment at the bottom in different colors. So like all of the genes right here are in this like green module, right? It actually turns out that if there's genes that have very little co-expression between other genes, they're not given any module, they're just assigned gray. So it looks like, I don't know, roughly like half the genes are just not even, they don't even end up in a module. Maybe they're lowly expressed in this particular cell type or something. So the two main outputs of this are the assignment of a gene to a module, and then something called the topological overlap matrix, which is actually what, it's like this weighted adjacency matrix, is they refer to it as topological overlap matrix. So that's the actual network. Okay, so there's a couple other things we want to do, which is we want to compute basically a score that summarizes the gene expression for the entire module. And this is something called a module eigengene. So, I mean, you can think about module eigengene pretty simply. If you have, let's say you have a module of 100 genes, you take the gene expression matrix just for those 100 genes, and then you perform principal component analysis on those 100 genes. Then you just take the first principal component, and that's the module eigengene. But we know for single cell, in principal component space is often confounded by batch effects. So our module eigengene function actually has an option to perform harmony batch correction, which gives you harmonized module eigengenes. So the module eigengenes function has this group.by.bars, which will allow you to do harmony batch correction on whichever variable you select for. And then finally, there's this function that computes the connectivity between the genes in each module. So it's just called module connectivity. This helps us to figure out which are the hub genes, the most highly connected genes in each module. Okay, so now like we can visualize the results of the module eigengene in the UMAP space. So here, each dot is a cell in the UMAP space, and it's colored by the module eigengene. So you can kind of tell like which clusters or which cells each of these modules are expressed in. And then we basically include this one function that just makes all of these different UMAP plots for whichever module you want. All it is this one. I mean, it's pretty much everything in this package is like one line of code, and it'll just do it for you. So we have the module feature plot function. It's kind of like the feature plot function from Seurat, but it plots. It basically just has this special like color scheme. If you haven't already noticed, because I didn't say it explicitly, but WGCNA assigns a unique color to each module. They're often kind of referred to by their color too. So you'll be like, oh, the purple module is like a synaptic transmission module or something like that. You can also just use straight up Seurat plotting functions to visualize the module eigengenes. So all you really have to do that is add it to the Seurat metadata. So there's like a tiny example, like all you have to do is take the Seurat metadata, and then you add the module eigengenes, and then like we can make a violin plot. Good thing Emmanuel's not here to see this. And then you can also make like a dot plot or whichever other function you want with Seurat. Okay, so now like aside from the Seurat visualizations, to actually visualize the co-expression network, we use under the hood, it's using this iGraph package, which is a cool package for doing network analysis and visualization in R. So like I was showing earlier, these are kind of like the classic like circle plots. These are common in a lot of different WGCNA papers. So we have a function just called module network plot, which it'll basically make a PDF that has one page for each module, where it's like the top 25 hub genes and their connections, and they're colored by their unique module color. There's other people have come up with a lot of different ways to visualize WGCNA networks. So we have this approach also, which is, I call this like a combined hub gene network. So, and it's from the function hub gene network plot. So you give it a Seurat object, and then you say like, I want 3 hub genes, and then I want 5 other genes. So for each module, it'll grab the top 3 hub genes ranked by connectivity, and then randomly sample 5 other genes. So the big genes that have a label are the hub genes, and then the small ones are just the other sampled ones. And then we plot it with a force-directed graph layout. And then, I mean, that's exactly what it is. So this one, I kind of plotted it and I'm like, this is like kind of, I don't know, it's kind of like still just a mess. And like, you have to go into like Adobe or something and pull apart all the genes to make, you know, to make them not overlap and all that stuff. Okay, Axel's asking, is it possible to color the edges or vary edge thickness based on the hub score centrality? Yeah, so the edge weight is actually scaled by the value between the 2 genes in the topological overlap matrix. So that's kind of like the edge weight is just co-expression between those 2 genes. And then, so because I'm always thinking about like, I don't know, what if you want to see like everything together? So I tried to run UMAP just directly on the topological overlap matrix. So it's just as a reminder, it's a matrix, you know, it's a square matrix with a value for each gene in the rows and in the columns. But I think that we can basically just take subset the columns to only include the top hub genes for each module. So we have this run module UMAP function where you tell it how many hub genes you want for each module, and then you give it like the regular UMAP parameters, like the distance and the number of neighbors, et cetera. And then you can just, you know, that actually runs quite quickly. And then you can just plot it with like ggplot or something, and then you get something like this, where each dot is a different gene, and it's the size is scaled by the intramodular connectivity. So the bigger genes will be the more connected in each module. And each gene is colored by its WGCNA module assignment. So just kind of looking at it, you can see that in the UMAP space, they are grouping together. So that's cool. And they're not kind of like, they're not overlapping like as much like this other plot, even though there's way fewer in this plot. So it looks to me like UMAP is a decent way to visualize the whole network. But like, okay, yeah, you know, the network has like edges as well as nodes. So like, how can we actually see the edges? So we include this module UMAP plot function to plot the whole network together. And you can imagine with all this different genes, there's a lot of different network edges. So we basically have a parameter that just plots like the top whatever percent of edges in each module. And you can also just do like a random sample of the edges as well. And in this plot, the color of the edge is actually, so it's gray if the genes are in different modules, and then it's just the module's color if they're in the same module. So for example, like, I don't know, if you see like in the red module over here, all the edges inside of it are red. So it's kind of a nice visualization. And you can actually see this animation I made for what does the UMAP look like when you vary the number of hub genes from, I think, 1 to 50. And interestingly, like overall, the structure is still like pretty similar. One of the modules kind of gets split apart, like this, I think it's like salmon or something. You can see it kind of getting split apart at the different parameters. But yeah, overall, it seems pretty robust to the number of hub genes that you give it as features. Okay, so kind of like if we remember like the very start of like the workflow WGCNA, we also kind of want to figure out like the function of each module. So we include built-in enrichment analysis functions. So all you have to do is the run enricher function to basically perform GoTerm enrichment analysis. So we give it a list of GoTerm databases, and then we tell it how many genes per module to test. So like I'm just giving it 100, which maybe that's too many. Maybe you just want like the top 25 hub genes. And then like whatever databases are in enricher. And then it actually runs pretty quickly, even though there's like tons and tons of overlap tests. And then you can get the raw output with this get enricher table function, or there's if you want to visualize the top results, you can use the enricher dot plot function, which makes this nice dot plot over here where it's showing the top one term for each module, just based on what we gave the function. And then it's colored by the unique module color, and it is the size is scaled by the enrichment value. So this will give you a basic idea of which biological processes each module are involved in. We can also compare the scWGCNA modules to cluster marker genes. So like, here's just a tiny bit of code to find marker genes with Seurat. It's just this find all markers function. Maybe it's familiar to you. It's going to give us marker genes that have a log fold change greater than 1. And then we can basically overlap with this table that comes straight from Seurat. We can like use this overlap modules DEGs function to compute if there is statistically significant overlap between the genes that make up a certain module and the genes that are marker genes from this marker gene test. And then you can also extract this table and do whatever you want with it, but we do include this module overlap bar plot function to visualize the like how much are these overlapping. And we're looking at the odds ratio here. So we can see that there is like an overlap in a lot of these. Okay. The other thing we kind of want to do, like this is often done in WGCNA, is like you usually do this kind of, I guess we see this a lot in machine learning. We have a training data set and a testing or a validation data set. So if there is like in your system of interest, if there's a publicly available data set that you want to compare to, you can run something called a module preservation test. And unfortunately, I also don't have that much time to explain what the test is doing, but there's this very long paper from 2011 called Is My Network Module Preserved and Reproducible? And they go through in excruciating detail all of the statistics involved in this test. So for example, we were using this Zhao et al. Human brain data set, and then we're going to basically project the modules that we discovered into my data set, and then we're going to test if they're significantly preserved. Okay, so to project the modules, it's basically just taking the module assignment from the reference data set, which in this case is the Zhao et al., and it's going to recompute the module eigengenes in the query data set, which in this case is the one for my paper. And once again, you can give it like the different batch correction variables for harmony and so on. So yeah, it's all it is one function, and then you can project the modules into a new data set. So you can see here, it's the same 23 modules, but now it's plotted onto the UMAP from the query data set, basically. And then all you have to do to compute the module preservation is run the module preservation function. I think this is the slowest function in WGCNA because it is based on like a permutation test. So it is running like, it's like running like iteratively over and over again for like a lot of iterations. So it might take like 3 to 4 hours to run. And then it gives you a lot of statistics. And then we have this function to make these module preservation plots. So on the x-axis is the module size, like how many genes. And then on the y-axis is the different module preservation statistics. So they kind of, I guess I'll show the next slide for like 1 second, but they give you like a lot of different statistics, and these are all described in the paper. But they have these 2 summary statistics for quality and preservation. So is like, this one is telling you like, is the network quality preserved? And the higher value means like, yes. And then there's these 2 cutoffs, right? So the dark gray is like, no, it's not preserved. The light gray is like, okay, it's a little bit preserved. And then the white is like, yeah, it's really preserved. So I guess the, like down here, like these 3 modules didn't really make the cutoff. And then these like 5 or 6 modules are like somewhat preserved, and these ones are very preserved. So you can use this analysis in a number of different ways. Like you can just use it kind of as reproducibility, but you can also kind of see like, is this module present in a different biological system? So like if you wanted to, for example, whoops. So like if you wanted to project your module into a spatial transcriptomics data, you can also use that with the same function. So here we're actually using the human Zhao et al. Reference, and we're using a mouse Visium spatial transcriptomic sample. So all we have to do to map to a different genome is also supply this function with a table called like a gene mapping table, where there's one column with the name in one species, the gene's name in one species, and one column with the gene's name in the second species. So yeah, and then you just tell it the name of the column, like HT38 MM10, and then voila, you end up with projecting the modules into the spatial data set in a whole different species, which is pretty cool. I didn't put the module preservation statistics here, but you can also see like, oh, maybe it's only preserved in one brain region or something. So there's lots of different questions you can kind of ask with that module preservation analysis. And then the last thing I'm going to talk about is just very quickly, we kind of include this analysis to do like motif enrichment analysis to see like which transcription factors are the target genes of different transcription factors enriched into your scWGCNA modules. So this is basically just 2 different functions. One is called motif scan, where it's going to take a position weight matrix list, which you can get from the TFBS tools package. So this is basically a list of the different binding motifs for, this one isn't just species specific, it's actually for like all vertebrates. And then you can scan the promoters of the actual promoter sequence for all of the genes in your Seurat object. And it'll basically tell you, like, is this motif found in this promoter? So it gives you like a matrix that's just a 0 or 1 for all the genes in your Seurat object, and then all of the motifs in your position weight matrix list. And then you can do this similar to what we've done before with like overlapping with the marker gene list. We can just do an overlap between the modules and then the target genes of these different motifs. So you can see on the right is a plot with this like module M1, the top motifs that overlap. So, and then like their odds ratio. We also plot the little like motif logo here, so you can see which ones they are. So this is kind of telling you like which transcription factors are potentially regulating each module. Okay, I think that's all I have. So in conclusion, go try out the package. Hopefully it works, because I don't think anyone's tested it yet, other than myself. And thank you so much for listening.
Does anybody have any more questions for Sam? Okay, Vincent has a question. Could you include clinical data as a variable?
So I didn't put it in this presentation, but there is it's one of the like vignettes that's a work in progress, is module trait correlation. So that would be kind of telling us if one of the modules is related to a certain clinical variable, that would tell you that. Like including like age, sex, disease status, as well as like the technical covariates. So like, what if your module is only influenced by a batch effect, or if it's like because of high mitochondrial gene expression or something, that'll identify which ones are like I don't know, they might just be like modules you don't really care about anymore because like it's driven by the technical variants.
So I have like a bunch of questions. It's
Okay.
more concern. It's more concern. Okay. So how big the data that you try to run it on your single cell WGCNA? And like how the runtime, how long did it take to like do the main part?
I have like one data set that's like 40,000 metacells and like 10,000 genes, and I think it ran in like 2 to 3 hours.
Okay, cool. So it's a little, it seems it's faster than the regular WGCNA, right?
I don't think it is, because it's using the same functions. They like,
okay.
the actual function to construct the network is the same. Well, the thing is, I always do a parallelize. So if you parallelize it, it's way faster. Like I do like 32 cores, and then it's like pretty fast. I don't know.
Can you parallelize on CPUs, or is it just GPUs? Because it's network stuff.
It's CPUs. This is not GPUs.
This is the, like, I think it's the like built-in function in the WGCNA that you can parallelize all your function. Yep. Okay, cool.
Yeah, it should maybe include like, the website doesn't tell you how long any steps run for, but I feel like I should add that.
No, I think no, I just asking because like, I don't know if you know or not, but most of the time I face this problem that I couldn't run like big data on the R or running it through the WGCNA. So I just curious how your package handles it.
Yeah, I think there's going to be an upper limit to how much it can handle, but I haven't like found it yet.
Okay, cool.
So this is a question that's kind of on the tail of Vincent's question. So you mentioned that you tend to run WGCNA on each sample and cell type combination. So what happens if your clinical variables are split based on those samples? So, you know, you have your disease samples and control samples, or maybe I'm just misunderstanding something.
Oh, so basically the Metacell step, I'll like go all the way back to that. Okay, so when you make the Metacell matrix, it's basically telling you like which cells are you allowing it to combine together. So like if I gave it nothing to group by, that means we don't retain any information about which cell type it came from or which sample it came from, because it's going to be potentially a mix, right? So if you want to retain this information for downstream use, you have to like specify. But
Got
then,
it.
like,
but you're not actually running WGCNA on the sample. It's just for the Metacell construction.
Yeah. So like then like I think we use the inhibitory neurons for this whole thing. So like there in the inhibitory neurons, there's metacells that come from all the different samples.
Got it.
Right?
Okay.
So they're all there together.
Thank you.
Yeah, there are different ways of like WGCNA is pretty modular, so you can kind of run it in many different ways and it's still valid. Like for example, you could make your modules in just your female samples, and then you can run the preservation tests in the male samples to see which ones are there. Or like you could make the modules in the disease condition and then test them in the control and so on and so forth. I don't know. People do all sorts of different stuff. But like the package should be flexible to do whichever kind of analysis you want. I hope. there any more questions? If not, we can just break.
Yeah, if there are no more questions, or if there are questions after the fact, Sam is on Slack. Go ahead and send him a message and try it out. And thank you so much for coming. See you guys soon.